Stockholm, Sweden, from the Karolinska Institute, and I see also from Pittsburgh. So you are coming from two places at the same time. He's our last speaker on graphene oxide interactions. Please.
Thank you very much, and I want to thank the organizers for inviting me. I'm very happy to be in this session. I think the talk that I plan to give today could also probably have been a good fit for the graphene session earlier today. But nevertheless, I'm going to focus on graphene oxide and interactions with primary human innate immune cells, specifically neutrophils and macrophages. And the work that I'm presenting has been performed in the frame of the flagship project, Graphene, which is a large European project, a 10-year effort. And within the flagship project, there is a work package consisting of some 12 groups or so dealing with health and environmental safety of graphene-based materials. Now, at the very beginning of the flagship project, we published this paper in Angewandte Chemie, where we tried to develop a classification scheme for graphene-based materials in order also to guide our toxicological studies and to allow us to do those studies in a more systematic manner. And briefly, what we are looking at are three parameters: the number of layers, and as well as the average lateral dimensions of the materials from nano to micron size. And the third parameter is the carbon to oxygen ratio. What I'm going to discuss today are essentially graphene oxides of small or large lateral dimensions. So we're going to compare these two. I want to give just a brief background based on the published data. These are not our studies, these are studies available in the literature. Namely, the following two studies, which are presented here, suggesting that graphene oxide triggers cell death in macrophages. And the suggestion from this study, so let me go back, from the study, the reference is at the top of the slide, is that graphene oxide triggers a TNF-alpha secretion, which leads to an autocrine necroptotic, that means regulated necrotic cell death. There is a second study also shown in this slide, also published in ACS Nano, where the lateral dimensions of graphene oxide were suggested to be important for this TNF-alpha production and other pro-inflammatory effects. And again, in both studies, there is a suggestion that TLR4 is involved, and also here, because this is a TLR4 inhibitor, which in this study prevented or suppressed the TNF-alpha secretion. Now, what I'm going to show you today is our data, which suggests that none of this is true. And I want to provide maybe a partial explanation also for this. Of course, taking into account that different cell models maybe have been used, different varieties of graphene oxide may have been used. But one key element is, of course, endotoxin contamination. And we heard a very nice presentation on this topic earlier in this session. What we have done in the flagship project is to evaluate graphene oxide samples for endotoxin. Just to make a long story short, we found that the conventional LAL tests, chromogenic LAL assays and so forth, are not applicable to graphene oxide. That is, we do see quite some significant interference with the LAL assay. We then developed, let's say, a variation on the macrophage activation test, which we call the TNF-alpha expression test. Basically using primary human macrophages, we incubate these with the graphene-based materials with or without polymyxin B, which is an LPS inhibitor. And now if we see TNF-alpha secretion, and if this is suppressed by polymyxin B, we can then conclude that this is a result of endotoxin contamination. If it's not suppressed by polymyxin B, it's likely to be an intrinsic effect of the material. And together with the lab of Kostas Kostarielos, also partners in the flagship, we also developed guidelines for let's say, sterile synthesis of graphene oxide. But essentially, we all know that endotoxin is present everywhere, so it's important to use endotoxin-free water, to use sterilized glassware, and so on. Now, using this endotoxin-free graphene oxide, we interacted the materials with primary human monocyte-derived macrophages. And contrary to some studies in the literature, we don't see a masking effect. We don't see graphene oxide aligning with the plasma membrane. What we do see is both for the small graphene oxide to the left and the large graphene oxide flakes, we see very nice cellular internalization. And you may appreciate that there are packages of graphene oxide like accordions compressed into vesicles within the cell. And also this is true for the large graphene oxide flakes. So we see internalization. We don't see any signs of toxicity. And this is up to 100 microgram per ml. Neither by ultrastructural features, by electron microscopy, or by Alamer blue assay, for instance. So large and small graphene oxide flakes in this cell model are both non-toxic. We then did the multiplex array to profile the cytokine and chemokine secretion. We used a commercially available panel of some 26 or 27 cytokines and chemokines. I focus here on one important example, namely TNF-alpha. And basically, as you can see, we don't see TNF-alpha production using endotoxin-free graphene oxide, either small flakes or large flakes. Our positive control is LPS. I give you one more example. We did the same experiment, the cytokine profiling, in primary human macrophages with or without LPS priming. So that means we pre-stimulate the cells with LPS to have a more activated phenotype. And we then found that certain cytokines are in fact induced. And here we're looking at IL-1 beta. Again, LPS is our positive control. What we now see is that in the presence of LPS priming, we see quite a pronounced IL-1 beta secretion, both for the large flakes and the small flakes. But note that there is no size difference. Both large and small flakes trigger IL-1 beta to a comparable degree. IL-1 beta is normally, secretion is normally a result of inflammasome activation. So switching now from primary cells, we used THP1 cells that were genetically deficient for various components of the NALP3 inflammasome, and the results are shown here. So the bottom line is that in standard, let's say standard THP1 cells, both LPS and small and large graphene oxide flakes triggered IL-1 beta. But this is completely abrogated in cell lines that don't express ASC, which is one of the components of the inflammasome, or NALP3 itself, or caspase-1, which is, of course, the enzyme which cleaves pro-IL-1 beta to produce mature IL-1 beta. Again, in this cell model, we also see no difference between the small and large graphene oxide flakes. So to summarize this first part of my presentation, we do not see any cytotoxicity for graphene oxide in primary human monocyte-derived macrophages, neither for small or large graphene oxides. These are nanoscale, these are micron scale in letter size. Again, using LPS-free graphene oxide, we don't see TNF-alpha production, which is contrary to the published literature. But we do see in LPS-primed macrophages that there is IL-1 beta secretion through the canonical NALP3 inflammasome pathway. However, these effects are size independent, again, in contrast to several other studies in the literature. But an important note here, and a non-trivial issue, is that we have to work with endotoxin-free materials, or we need to control for endotoxin if we are going to use immune-competent cells such as macrophages. And this is perhaps even more true for neutrophils, and I will discuss this in the remaining few minutes of my presentation. Neutrophils, as you all know, are specialized and professional cells of the innate immune system, specialized in killing bacteria and fungi. And they can do so essentially through two different pathways, either intracellularly or extracellularly. So neutrophils can internalize microbes, which are then proteolytically or oxidatively destroyed within the phagosomes inside the neutrophil. Alternatively, neutrophils can produce so-called neutrophil extracellular traps, or nets. Now, nets are essentially a network of nuclear chromatin, which is expelled from the neutrophil. And this nuclear chromatin network is studied with granule proteins, including neutrophil elastase, myeloperoxidase, etc. Also, histones are present. And in these extracellular traps, the microbes are trapped and destroyed. And we have shown here that we can detect neutrophil extracellular traps by staining for extracellular neutrophil elastase. And shown in blue are the cell nuclei. So we asked the question whether graphene oxide is sensed, in a manner of speaking, by neutrophils as microbes. In fact, what we have found is that graphene oxide triggers neutrophil extracellular traps in a size-dependent manner. We use two different assays to measure nets, to quantify nets, the neutrophil extracellular traps, either by measuring extracellular DNA or extracellular elastase activity. And in white are the small flakes, in black are the large flakes. So there's a dose-dependent and size-dependent induction of nets. These, by the way, are the size distribution data for the small flakes, which are some 80 to 100 nanometers in diameter, and the large flakes some 8 to 10 microns in diameter. We also have begun some investigations to look at the mechanism of net induction. And we asked the question, is the NADPH oxidase important? It's well known for PMA, for instance, which is commonly used to trigger nets, that this acts by activation of the NADPH oxidase. If we add DPI, which is an NADPH oxidase inhibitor, we can reduce the amount of nets. Now, what we find is that for the large graphene oxide flakes, the NADPH oxidase inhibitor has no effect, whereas for the small graphene oxide flakes, there is an inhibition. So there is a difference between large and small flakes. Just for comparison, it may be hard to discern here, but these are electron microscopy images of neutrophils which have internalized small graphene oxide flakes encircled here. Whereas if in the case of neutrophils, we actually don't see much internalization. Instead, we see that graphene oxide aligns with the plasma membrane, and even with some imagination, you might say that there is a stripping of the membrane, and the graphene oxide seems to strip off the plasma membrane from the cell. So the behavior here is quite different, and we may account for these differences. But we are continuing to investigate this mechanism. This is a scanning electron microscopy image just to show you what the nets actually look like. This is a neutrophil which has produced so-called neutrophil extracellular traps. You may see the chromatin network here. And this is a graphene oxide flake, a large flake, which has triggered the net induction, and after which the graphene oxide itself is trapped in the nets. Now, as I told you before, nets contain granule proteins, including myeloperoxidase. So we asked the question, can the nets actually degrade graphene oxide extracellularly? And to do this, we triggered neutrophils with PMA, which allows us to trigger net formation. We then purified the nets from the cells. So we have purified nets. We incubate these with graphene oxide. And in fact, using Raman spectroscopy, we can look at this characteristic Raman spectra, and we do see a time-dependent oxidation, which is an indication of degradation of these materials in purified nets. So this is acellular degradation of graphene oxide with time. And we know that it's myeloperoxidase dependent because in the presence of the MPO inhibitor, there's no oxidative damage. Finally, we also asked whether activated neutrophils, which are activated with the agonists shown here, can also degrade graphene oxide. So activated neutrophils will release myeloperoxidase, and we then asked the question, will this degrade graphene oxide? Again, using Raman spectroscopy, we see both for small flakes and for large graphene oxide flakes at the bottom here, a time-dependent degradation. And perhaps not surprisingly, it's more efficient, or more rapid, I should say, for the small graphene oxide flakes. So this is the first demonstration of neutrophil-mediated degradation of graphene oxide. To summarize then, in contrast to macrophages, where we did not see size-dependent effects, and actually no toxicity either. For neutrophils, we do see that graphene oxide triggers nets in primary human neutrophils. This is size-dependent. And we also see that these nets, which contain myeloperoxidase, can actually digest or biodegrade graphene oxide. Moreover, as I showed in the last slide, or the previous slide, that the activated neutrophils can also digest graphene oxide, and this is also an extracellular event due to the release of myeloperoxidase. So overall, I mean, our innate immune system has evolved to protect us from foreign intrusion, microbes, fungi, etc. Based on these studies, and I apologize for going so quickly through these data, but based on these studies, we may speculate that the immune system can also sense two-dimensional objects, which I think is rather fascinating, and actually use very similar mechanisms, in this case net production and digestion, to destroy, as it were, these materials. Finally, let me acknowledge the people who did the work, the people in my lab, in particular Surav Mukherjee, shown here, who really has been responsible for all the graphene oxide-based work in our lab. Other collaborators in Stockholm. I also want to highlight Kostas Kostarielos and his postdoc Nevs Lozano, who provided us with the endotoxin-free graphene oxide, and our collaborators here in Switzerland in St. Gallen, who were involved in the endotoxin studies. And the work was funded by the EU Graphene Flagship project. Thank you very much.